are talking about mitosis and meiosis. And today I will discuss to you what are the process of mitosis and meiosis. But before that, let we know what is mitosis and what is meiosis first. Mitosis is a process where a single cell divides into two identical daughter cells. During mitosis, there's only one cell, and once it divides, they form at least two identical cells. The major purpose of mitosis is for growth and to replace worn out cells. Next one is what is meiosis? Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. These cells are our sex cells, sperm in males, eggs in females. Meiosis produces our sex cells are called gametes. Again, eggs in females and sperms in males. So now let we know what are the process of mitosis. The process of mitosis is used for growth and repair within an organism, also for asexual reproduction. There are four main stages to mitosis. That's called prophase, metaphase, anaphase, and telophase. Although the process has been divided up into these stages, the process of mitosis is actually continuous. First, what is prophase? Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus known as chromatin. Next one, what is metaphase? During this stage, the chromosomes move towards the equator of the spindle, attaching themselves horizontally by their centromere to the spindle's filaments. The chromatids then pull slightly away from each other at the centromere towards the opposite poles of the cell. The third one is the anaphase. Anaphase is the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. Lastly, what is telophase? Telophase is the final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of the parent cell into two identical daughter cells. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. So next one, let me know what are the process of meiosis. Meiosis happens in a two-stage process of cell divisions that produces four haploid cells from one diploid cell. The process begins from a single dividing nucleus that is composed of two sets of replicated chromosomes that contain two sister chromatids, nuclei that consist of a single set of unreplicated chromosomes. These are the phases of meiosis 1, the prophase 1, metaphase 1, anaphase 1, and telophase 1. First, the prophase 1. This is a starting cell is diploid, homologs, chromosomes pair up and exchange fragments that is crossing over. Next is the metaphase 1, where homolog pairs line up at the metaphase plate. The third one is the anaphase 1. Homolog separates the opposite ends of the cell, sister chromatids stay together. And the last one of phases of meiosis 1 is the telophase 1. This is newly forming cells are haploid. Each chromosome has two non-identical sister chromatids. Next is the meiosis 2. During the second round, the sister chromatids separate, making haploid cells with non-duplicated chromosomes. In meiosis 2, there's a prophase 2 and metaphase 2 and phase 2. First, the prophase 2. Starting cells are the haploid cells made in meiosis 1, chromosomes condensed. Next is the metaphase 2, the chromosomes line up at the metaphase plate. And lastly, the anaphase 2, the sister chromatids separate to opposite ends of the cell. So that's all. Thank you for listening.